Kingilla is also belonging to Neissereaceae family, and both both oxidase and nitrate positive organism. And in this group, uh, Kingilla dendrificans is very important because causing the endocarditis with non-hemolytic nitrate positive, and also DNA is positive and required thyre martin to grow. Kingilla kingi is also very important organism in this group that is causes the osteomyelitis, septic arthritis, septicemia, and a beta hemolytic with nitrate negative. Another Kingilla aurelis is also nitrate negative non-hemolytic, but found in dental plug. Another organism in Hag group is a Cardiobacterium, which belong to the Cardiobacteriaceae, and this organism is oxidase positive but nitrate negative, and also indole positive, and also produce acid from the glucose and sucrose, can grow on SBA chocolate and do not grow on the McConkey microaerophilic condition. It's a pelomorphic bacilli with the swollen ends. The most important pathogenic organism in this group is a Cardiobacterium Haemophilus, which cause endocarditis, and this organism have alkaline phosphatase reaction positive. Villosan is alkaline phosphatase reaction negative. In Cardiobacteriaceae, another important organism is a Sothonyla indulgens, and that is plum gram negative bacilli, slow growing, producing pitting in agar, oxidase positive and indole positive. Produce the glucose and sucrose, acid from glucose and sucrose, and alkaline phosphatase reaction negative, just like the Cardiobacterium velorium, which also alkaline phosphatase reaction negative, but that do not produce the pit on media like a chocolate. Another organism in the Bacteriaceae is a Chromobacterium volucum. It's from the name indicate that the organism is capable of producing the pigment. The organism is also capable of causing diseases like skin cutaneous lesions, diarrhea, UTI, deep violet pigmented pigments, that is called violusine. These pigments may interfere with the oxidase reagent and confuse the test. The ID for this Chromobacterium volucum is a violusine oxidase, urea, indole, and arginine dehydrolase, and acid production from the glucose is positive. In Hack group, there is one species that is only positive to nitrate, and that is Aggregatibacter, which belong to Pasteurolaceae family. Again, it's found in the human oral cavity, and due to the human bite, this infection cause, just like Acnella cordon, which is also due to the human bite. But this organism is only nitrate positive, which differentiate from Acnella. Another is the most important organism in this group is Aggregatibacter actinomyceticotans. That is a predonatal disease, endocarditis, gram-negative rods, filamentous form, SBA, 48 to 72 hours required, and they produce the star-like colony, a pipting of agar. Aggregatibacteria family also include the Aggregatibacter Haemophilus and Aggregatibacterius Sonegus, and these both organisms require the V factor NAD for their growth. And these bacteria before classified in the Haemophilus group, but the Haemophilus is oxidase positive and catalase positive. So the Aggregatibacteria is only nitrate positive and produce some acid from glucose. And then we will discuss the one bacterium which is most important in this group, and that is Streptobacillus minoliformis, which is the only glucose positive species in the whole Hack group. And this organism is very important organism because this organism is normally found in the respiratory tract of rats and cause the streptobacillary rat bite fever, which this infection can cause by the ingestion of a water milk, and also this is called Havery Hill fevers. Epidemic arthritic erythema is also another name of Streptobacillus minoliformis. This is polymorphic gram-negative bacilli with the area of swellings filamentous. The growth requirements are blood, serum, and aesthetic fluids. The interesting feature of this organism is they produce the L form on SBA plate, and in the broth medium grow towards the bottom of the tube and form some breadcrumb or cotton ball like appearance in the broth medium. And same on the gram stain. The gram stain, they also show the cotton ball like appearance, and that cotton balls are associated with one another by some threads like appearance. That is basically Streptobacillus minoliformis, and people usually confuse by the stain artifacts, but this organism you need to check whole slide and check whether it is Streptococcus minoliformis or some stain artifacts. They can grow on SBA with the smooth, glistening, and fried egg appearance, which is very important identification. The organism is catalase positive also, and produce weak acid from glucose and maltose. But in this group, we classify its glucose positive organism. But another feature of this organism is catalase positive, which is very important. Among the hack group, there is another organism that is called Capanocytophaga. This is a dog oral flora, and capability of producing acid from the glucose and sucrose. It's a thin gram-negative rods with pointed ends, just like a Streptococcus pneumoniae, with the gliding motility and swarming on chocolate. Tiny pinpoint yellow-orange colony with 5 to 10% carbon dioxide or anaerobic condition is required for this organism. In this group, Capanocytophaga, there is three main species: the Capanocytophaga hemolytica and Capanocytophaga sputigina. Both are nitrate positive, and Capanocytophaga granulosa is acetylcholine positive. It's an oral cavity, can cause the dental plug, subguinal plug, wound, blood, and respiratory secretion. Another organism in which is quite similar to the Capanocytophaga is Diagomonas. Diagomonas is non-motile, and if you see the Capanocytophaga is motile with the gliding motility, and Diagomonas is a cocobacilli with strawberry-like odor. So another identification for this organism is ONPG positive, along with glucose and sucrose. So the Capanocytophaga is only glucose and sucrose positive, ONPG negative, and this organism can be isolated from the blood and other body fluid. Gallbladder wounds produce acid from the lactose, sucrose, xylose, and this organism has some subspecies, that is Desogomonas capanocytophagiod and Desogomonas giladi, Desogomonas musi, Desogomonas hofstadi. Another organism in the Hack group, which is most important, is Haemophilus. Haemophilus is oxidase positive, catalase positive, but nitrate negative organism, and Haemophilus required the special requirement for the growth, that is X factor, which is hemine, V factor, which is nicotinamide adenosine dinucleotide. This is also called prefer bacillus. Requires grow on the chocolate agar, because chocolate agar contain both X and V factor. And on SBA, the organism required some NAD producing bacteria like Staphylococcus aureus, and this process is called cytolysis, when you can grow the Haemophilus species on Staphylococcus aureus growth. The organism cause the epiglottitis, endocarditis, meningitis. If you see the chart, you will find that Haemophilus influenzae required both X and V factor, but can produce acid from the glucose also. Para-Haemophilus only required the V factor for the growth, but can produce acid from the glucose, sucrose and mannose. And if we see the H. hemolyticus, that is just similar to the influenzae, required both X and V factor, but also produce hemolysis. So which is the only differentiation between the influenzae and hemolyticus. Aegypticus is like a Haemophilus, but do not have any ability to produce acid from the glucose set. Both required X and V factor. By Aegypticus, Dusra is causing the STD, and they required only hemine X factor. And para-hemolyticus is similar to the para-influenzae. They require X factor for the growth and show the hemolysis, but do not have any ability to produce acid from the glucose. This is a slow growing bacteria. Haemophilus influenzae is categorized as a biotype 2 agent, and Haemophilus aegypticus is known as cocci bacillus, and their indole and xylose is negative. It causes the pink eye conjunctivitis. Both Haemophilus dusrai, which cause the STD, both takes 5 days at 30 to 33 degrees centigrade to appear the colony on plates. Gram-positive bacilli. First we describe in the gram-positive bacilli the spore forming. The spore forming bacteria among this group is first we check whether it is beta hemolytic or motile. If it is beta hemolytic and motile, that is Bacillus species, boxer shape and causing the food poisoning. If it is not motile and also not beta hemolytic, then we go for the Bacillus anthrax, which is a boxer shape and which is most pathogenic organism in this group, and that can cause the skin that can cause the mucosa head-like, that is the mucosa head-like colonies, and cause the black ulcer on skin. And if we check that the gram-positive bacilli is not spore forming, then we check for the branching. If there is a branching and it is gram-positive, then we go for the Nocardia species, which is beaded branches, change, partially acid fast, immunocompromised. Patient suffer from the infection. They can be serious pulmonary dissemination. If there is no branching, then we go for the catalase. If the catalase is positive, then bilirubin and motility. If bilirubin and motility is positive, then we go for the Listeria monocytogene, which is neonatal and immunocompromised neonatal pathogen, and some immunocompromised also suffer from septicemia and meningitis because of this, and intracellular organism and cell mediated immunity is also required to fight. They show umbrella growth in motility tube agar at room temperature, but not at 35 degrees centigrade, often found in the dairy products. If we have gram-positive rods and that catalase is positive but non-motile and bilirubin is negative, then we go for the Chironibacterium diphtheria. That is the most important pathogen, and they basically on the gram stain show the Chinese letter morphology and metachromatic granules with the Lufer methylene blue medium, and black colonies with the brown hollow on the tendrils, holy agar, or black colony only on cysteine tolerate agar, which is also holy agars, mostly cause infection in children. If the catalase is negative for gram-positive bacilli, then we go for H2S production. If it is positive, then it is Ascp, which is you can see in the agar motility tube opposite to the Listeria monocytogeneum. It shows the brush-like appearance, and Listeria show the umbrella. You can say the umbrella is inverted position, so it is quite opposite to Listeria monocytogene. And if H2S production is negative, then we go for Lactobacillus species and Gardnerella species. Gardnerella vaginalis, the clue cells are identification marker for this organism, which is from high vaginal swab. Further, if we see the catalase positive bacilli, that is catalase positive, gram positive bacilli, and motility and biologically negative, then we can differentiate it by some other classification, which is alpha, beta, and non-hemolysis. Alpha hemolysis Corynebacterium that required Holley-Zager, as we explain, have black colonies with the brown halo in the center, and beta hemolytic in this group is Arcanobacterium hemolyticum, that is positive for reverse cam test, and Arcanobacterium pyogenes that ferment the xylose, and Arcanobacterium bernardi that ferment the glucose, and non-hemolytic is Rothia and Gardnerella, as we explained. Now we will explain the Corynebacterium diphtheria in detail. If we see, Corynebacterium have so many species. The first one is Corynebacterium xerosis, that is glucose, that end product of glucose is lactic acid in identification. Another is Corynebacterium amyloificum, that glucose end product is propionic acid. And the two other organisms, that is Corynebacterium pseudotuberculosis and Corynebacterium ulcerin, they both are positive for urea in reverse cam. C. ulcerin ferment glycogen, while C. pseudotuberculosis does not. So this is the only identification difference. The Corynebacterium genicum is a resistant species because this organism required the twin AT for their supplement for the growth, and oxidize the glucose and sucrose and sometime maltose. C. uroliticum is a resistant species, and urine pH become alkaline if this organism is present in bladder, and in the urine also contain the strowit crystal, which is also identification marker or diagnostic marker for this organism. They can hydrolyze the urea and convert pH to alkaline and cause the UTI. Corynebacterium mcnigili is causing the eye infection, ferment the mannitol, and Corynebacterium regili hydrolyze the urea within 5 minutes at room temperature, and also ferment the maltose. This Corynebacterium entire family we can identify by another method in the clinical laboratory by putting the phosphomycin 100 microgram disc on a fleshly subculture isolate, and all of these organisms, all of this family Corynebacterium is actually resistant to phosphomycin 200 mg disc, so the growth will be appear on the. Another test is LX test, is in which we 10 microgram disc is used, and we see the lysogenic conversion. There is another bacteria that is very important on clinical point of view at microbiology laboratory, that is Legionella. Legionella is found in water system like cooling system, hot tub, low level chlorine water. Legionella pneumophilia and Domophi, and but mainly the Legionella pneumophilia cause 90% of infection, and the main transmission is through aerosol. It's aerobic organism, not visualized on gram stain, but gram negative rod, thin faint staining or non-sporing. This organism have acid fast, modified acid fast stain is required for this organism, and also this organism required L-cysteine for the growth. Also Francisella tularensis also required L-cysteine, depending on L-cysteine, so maybe sometime misidentify. This organism evaporate hydrolysis is positive, required 3 to 5 days 5 degrees centigrade at carbon dioxide, microaerophilic. And Pneumophila, as we explained, the serotype 1 is responsible for 90% of pneumonia. And the organism required iron under 5 degrees centigrade to enhance the growth. Gram-negative diplococci, caffeine bean shape. Gram-negative diplococci are majorly Neisseria gonorrhea, Neisseria meningita, Neisseria lectima, and Moraxella catarrhalis. This Neisseria gonorrhea is a very fastidious organism, and they required some extra requirement. And if we identify these organisms, we will identify on the basis of biochemical test. The organism is oxidase and catalase and glucose positive. And another unique feature of Neisseria gonorrhea is superoxal positive. Neisseria meningitis is also oxidase catalase positive, but it also ferment the glucose and maltose. But Neisseria lectimica is just like Neisseria meningitis, positive for oxidase catalase glucose maltose, also positive for lactose or ONPG and rhosis beta galactosidase enzyme, that is also positive for the Neisseria lectimica, while Moraxella catarrhalis is only positive to glucose to oxidase catalase, and the unique property is a DNase positive organism. Neisseria gonorrhea grow all other organisms can easily grow on the chocolate, but Neisseria gonorrhea required MTM, Martin Lewis, New York, agar, and this organism is diplococci, but it is not encapsulated, so it's very fragile in the gram stain. You need to mostly found from the male's urethral discharge, and you don't need to heat fix the stain, otherwise the morphology will be disrupted. Gram negative cocci, we are explaining Neisseria, and that is cholesterol sensitive, oxidase positive, catalase positive, but DNA is negative. Neisseria is a kidney bean shape, and the two major pathogen is Neisseria gonorrhea, which is non-capsulated, and Neisseria meningitis, which is capsulated. Neisseria gonorrhea have concave end, nitrate reduction negative, ferment only glucose, superoxal test is the unique property, that is positive. Neisseria species, all other Neisserias are negative for superoxal, only gonorrhea is positive. Require the capanophilic atmospheric modified Tyre-Martin agar, which contain the colistin, vancomycin, nystatin, and citron cotrimoxazole. And this Neisseria meningitis, which is hockey pock colonies, flat ends, nitrate reduction positive, ferment maltose and also glucose. In this species, the Neisseria lectimica, which are able to ferment the lactose, and Neisseria silica, which are able to ferment the sucrose. And Neisseria silica is cholesterol sensitive, but lectimica is cholesterol resistant, which differentiate lectimica from the entire group.